CIDER, a novel method for inferring causal links or subgraphs corresponding to a specific phenotype or label, addresses the challenge of causal subgraph inference from measured data. Unlike existing graph neural network (GNN) explainers, which often provide associative insights, CIDER aims to provide causal insights through the implementation of both counterfactual and diffusion mechanisms. This model-agnostic and task-agnostic framework generates causal explanations via counterfactual-invariant and diffusion process, yielding both causal subgraphs due to counterfactual implementation and reliable causal links due to the diffusion process. The proposed approach formulates an inference task that generatively provides two distributions, enabling the identification of causal subgraphs and links. By leveraging counterfactual and diffusion mechanisms, CIDER overcomes the limitations of existing GNN explainers, which often rely on associative insights. The counterfactual implementation in CIDER allows for the identification of causal subgraphs, while the diffusion process provides reliable causal links. The CIDER framework consists of two primary components, a counterfactual module and a diffusion module. The counterfactual module generates causal subgraphs by analyzing the differences in the graph structure between the observed and counterfactual scenarios. The diffusion module, on the other hand, provides reliable causal links by propagating information through the graph structure. The effectiveness of CIDER is demonstrated through extensive experiments on various benchmark datasets, showcasing its ability to identify causal subgraphs and links with high accuracy. The results highlight the importance of considering both counterfactual and diffusion mechanisms in causal subgraph inference, providing a novel approach to addressing this challenging problem. CIDER, or Counterfactual-Invariant Diffusion-based GNN explainer, is a novel framework designed to identify causal links or subgraphs with respect to sample labels based on measured data. This approach addresses the limitations of traditional association studies and observational causal methods, which failed to provide causal explanations. By modeling the graph as a diffusion process, CIDER enables the explicit quantification of each subgraph's causal strength in a counterfactual manner, reducing the effects of unobserved confounders and providing a more reliable approach to causal inference. Graph neural networks, GNNs, have emerged as a powerful tool in handling graph-structured data. However, existing methods, such as graph neural networks explainer, only provide explanations at a model level, failing to answer the critical question of which part of the graph results in a specific label. Previous approaches, such as those proposed in 1, 2, 3, and 4, have limitations, including ignoring the dependence between edges, using greedy algorithms, or reconstructing subgraphs with no causal links. CIDER overcomes these limitations by training a diffusion-based model to divide the whole graph into one causal subgraph and one spurious subgraph. This framework can answer both, which part of the graph causally affects a sample's label, and which part of the graph results in the trained GNN's output. The authors evaluate CIDER on both synthetic and real-world datasets, demonstrating its superiority over state-of-the-art methods in identifying causal links and subgraphs. This approach has significant implications for advancing our understanding of causal relationships in graph-structured data. CIDER, a novel framework, is illustrated in Figure 1, comprising a conceptual illustration and a framework. Given a graph with label Y, CIDER outputs two subgraphs: the causal subgraph, which causally affects the label, and the spurious subgraph, which does not affect the label. Notably, CIDER directly predicts the distributions of these two subgraphs, enabling the estimation of causal strength, defined as each subgraph's contribution to the graph's label. The CIDER framework, shown in Figure 1b, involves training a graph neural network, GNN, solely on measured high-dimensional data. This is achieved by implementing intervention and counterfactual-invariant to train a diffusion model, which directly constructs the distributions of the causal and spurious subgraphs. CIDER provides a causal explanation from a subgraph to a label or phenotype, leveraging the capabilities of GNNs in handling high-dimensional data and the novel training approach utilizing intervention and counterfactual-invariant. Furthermore, if the graph is not explicitly given, CIDER can utilize measured data X to construct a graph, for instance, through correlation analysis, enabling its application to a wide range of scenarios. CIDER, a novel method for inferring interventional causality in graphs, is presented. This approach utilizes a variant inference task to predict the distributions of two subgraphs, the causal and spurious subgraph within a graph. By employing a VGAEBASED framework with two channels, CIDER can differentiate between these subgraphs and quantify their contributions to the graph's label. The method also adopts a diffusion process to enhance its inference capability, resulting in a sparser and more reliable causal subgraph distribution. The theoretical results of CIDER demonstrate its ability to infer the causal subgraph against an output label or phenotype of a sample based on interventional causality. The method defines a random graph as G equals X, V, E, where E and V represent the numbers of edges and nodes, respectively. Each node has a d-dimensional feature vector, and X is a V times D matrix representing the measured data of a sample or graph. The goal is to derive the conditional distribution of the edges, P, E, X, V, from the data, which is simplified as P, E. CIDER's performance is evaluated through comprehensive experiments on both synthetic and real-world datasets, including single-cell RNA-seq datasets. The results indicate that CIDER outperforms state-of-the-art methods. Additionally, a framework is proposed to use CIDER to analyze single-cell RNA-seq data, illustrating its efficacy and applicability in real-world scenarios. The code and data used in this work are available in the supplementary material. A novel approach to causal graph explanation is presented, leveraging a two-channel variational graph autoencoder, VGAE, to infer the causal subgraph from an original graph. This method aims to identify and explain the underlying causal factors contributing to a specific label or task in graph-structured data. The proposed model is evaluated on both synthetic and real-world datasets, demonstrating its effectiveness in graph classification tasks. The resultant causal subgraph EC is derived from the original graph E through a series of transformations and interventions, while the remaining non-causal subgraph S consists of the residual components of the original graph after extracting the causal subgraph. By employing a two-channel VGAE, the model simultaneously infers the distribution of the causal subgraph P, EC, and the conditional distribution of the non-causal subgraph given the causal subgraph P, S, EC. Interventional causality is a key concept, defined as the relationship between the distribution of the resultant causal subgraph and the label Y, captured by the equation P, Y, CF, S, equals P, Y, EC. This result indicates that the converged EC is the causal subgraph for label Y, where S is the remaining non-causal subgraph among the original graph E. Computational results on benchmarks compare the proposed model with baselines such as GNI planar, Orphic X, GEM, and PG explainer. The model is evaluated in two parts: traditional GNN explaining experiments, focusing on the task GNN model's output without considering labels, and causal explanation experiments, which consider labels and aim to explain underlying causal factors contributing to the task. The proposed model demonstrates effectiveness in identifying and explaining the causal subgraph from an original graph, contributing to a better understanding of relationships between variables in complex systems. The two-channel VGAE approach enables simultaneous inference of causal and non-causal subgraphs, allowing for a more comprehensive understanding of graph-structured data. CIDER's performance is evaluated on both synthetic and real-world datasets, showcasing its effectiveness in complex pattern recognition challenges. In the synthetic BA2 motif dataset, CIDER achieves nearly 100% accuracy in classifying graphs based on attached motifs, demonstrating exceptional performance. This dataset, comprising 1,000 randomly generated Barabasialbert graphs with added motifs, presents a binary classification challenge that CIDER addresses with remarkable precision. Moving to real-world datasets, the evaluation focuses on identifying the parts of the graph that causally contribute to the label or phenotype. A modified loss function, L, is employed to train a graph neural network, GNN, to predict the ground truth of the labels. The results, presented in Table 1, demonstrate CIDER's superior performance compared to baselines such as GNI planar, PG explainer, GEM, and Orphic X. CIDER achieves higher accuracy in explaining phenomena on various real-world datasets, including MUTAG and NCI1, with different sparsity levels. CIDER's attention loss, C, is formulated as F, S, Ns, where S is a subset of edges, and Ns is the node set. This enables the model to capture the causality underlying phenomena in complex graphs. By requiring fewer edges to predict the ground truth, CIDER reinforces its suitability for addressing real-world data structure demands. The approach provides high explanatory power, improving upon existing methods by integrating graph context and explaining complex patterns in graphs with high accuracy and efficiency. The CIDER method is applied to real-world RNA-seq datasets, including COVID-19 single-cell RNA-seq data and AML bulk RNA-seq data, to demonstrate its adaptability and utility. By simultaneously elucidating both causal genes and cell-type networks, CIDER facilitates the identification of underlying biological mechanisms at a molecular level. The COVID-19 dataset, comprising 8,361,000 single cells from the blood of 124 individuals in 3 different states, is used to evaluate CIDER's performance. Initially, a graph neural network, GNN, is employed to classify the COVID-19 gene network, achieving an accuracy of 0.85. After applying CIDER, the network is significantly streamlined to 30 edges, while maintaining a classification accuracy of 0.82 with the same GNN. This result indicates that CIDER preserves approximately 96% of the performance in deducing genogene causal subgraphs, while substantially reducing the network's complexity for inference tasks. CIDER also manages to retain 91% of the performance in identifying cell-type causal subgraphs, demonstrating its efficiency in network sparsification without significant loss of accuracy. To further elucidate CIDER's effectiveness, the top 1% of highly variable genes, 375 genes, in the dataset are refined, and the top 30 genes with the highest causal association scores are selected for biological analysis. These genes are analyzed using tools like David and String database, revealing 3 independent subnetworks in the string network. One large subnetwork comprises genes such as CYBB and the S100 protein family, while the other 2 subnetworks are smaller, consisting of the RPS family of genes alongside FTL, and the standalone gene COTL1. These subnetworks potentially indicate distinct network functions, which will be further investigated using other analytical tools. The isolated COTL1 gene may have a unique, yet unknown impact, with previous studies reporting its role in the immune response and involvement in the innate immune system. Single-cell RNA sequencing analysis using the CIDER method identified 30 key genes related to COVID-19. These genes were divided into 3 distinct clusters based on known and predicted protein-protein interactions, revealing potential unique functions and interactions within the molecular network. Notably, KEGG pathway enrichment analysis showed that the key genes were significantly enriched in pathways related to ribosomes, apoptosis, tuberculosis, and COVID-19, suggesting a direct connection between the key genes and disease mechanisms. 4 key genes, including CYBB, RPS3, RPS18, and RPS27, overlapped with the COVID-19 pathway. CYBB, a major component of the phagocytic cell's microbicidal oxidase system, is implicated in the production of excessive reactive oxygen species. Gene ontology enrichment analysis revealed that the key genes were primarily involved in immunity-related processes, including inflammatory response, neutrophil chemotaxis, innate immune response, and apoptotic process. UniProt keywords enrichment analysis further supported the role of the key genes in immunity-related processes, with keywords such as innate immunity, immunity, and inflammatory response being significantly enriched. The results suggest that the CIDER method is effective in identifying COVID-19 related genes and highlighting their potential prognostic value for severe cases. The downregulation of COTL1 protein in the airway mucus of COVID-19 patients, as previously reported, is consistent with the findings of this study. Overall, the analysis provides insights into the molecular mechanisms underlying COVID-19 and identifies key genes that may be involved in the disease process. The exacerbation of clinical outcomes in severe COVID-19 infections has been linked to the overactivation of NOX2, associated with CYBB, suggesting a role for NOX2 as a mechanism favoring thrombotic-related ischemic events. The ribosomal protein family, particularly RPS3, RPS18, and RPS27, has been identified as a potential inhibitor of COVID-19 replication and a critical component of the COVID-19 pathway. RPS3, a hub gene in COVID-19, has been highlighted as a potential therapeutic target, as it influences both host and viral mRNA translation and stability. Gene ontology and uniport keywords analysis revealed that the majority of genes were enriched in immune-related keywords, supporting the effectiveness of the CIDER method in selecting COVID-19-related genes. The S100 calcium-binding protein family was identified as playing a role in inflammatory conditions in COVID-19 patients, with potential applications in predicting disease severity. These genes have a closely related function with CYBB, potentially activating CYBB in the immune response against COVID-19. Uniport keywords ligand analysis highlighted calcium and lectin as key terms, with literature suggesting that COVID-19 critically impacts calcium balance and serves as a biomarker of clinical severity. Hypocalcemia has been strongly positively associated with COVID-19 severity, and calcium channel blockers have been shown to reduce mortality from the disease. Lectin has also demonstrated efficacy in combating and treating COVID-19. The CIDER method effectively filters and identifies key genes with causal associations with COVID-19, unraveling intricate networks and pathways that offer a deeper understanding of the disease's pathophysiology and contribute to identifying potential targets for therapeutic intervention. The CIDER method was applied to the TCGALAML dataset, which includes gene expression profiles from 179 individuals with acute myeloid leukemia. The analysis used only the samples labeled as adverse and favorable. The gene expression data was cleaned, and the original gene network was constructed using strain and correction networks. The CIDER methodology was then applied to identify and refine the top 1.5% of highly variable genes from the original network, resulting in the selection of the top 51 genes with the highest causal association scores, maintaining an 88.6% accuracy. The analysis of the TCGALAML dataset demonstrates the effectiveness of the CIDER method in identifying key genes with causal associations with disease. The method's ability to filter and refine genes based on their causal association scores enables the identification of potential therapeutic targets and contributes to a deeper understanding of the disease's pathophysiology. The research paper presents a comprehensive analysis of gene networks related to acute myeloid leukemia, AML, using the CIDER tool. This analysis identified 51 key genes, which were then divided into two distinct clusters through network subgraphs generated by strain. These clusters indicate potential unique functions and interactions within the molecular network related to AML and ribosome. The study also utilized KEGG pathways to identify enrichment in pathways related to myeloid leukemia, pathways in cancer, and cancer-related signaling pathways. This enrichment suggests a direct connection between the key genes and the leukemia mechanisms. Furthermore, gene ontology biological process enrichment analysis was conducted for the identified key genes. This analysis revealed enrichment in various biological processes such as regulation of apoptotic process, regulation of programmed cell death, cytoplasmic translation, positive regulation of signaling, phosphorylation, cellular metabolic process, and cell activation. Additionally, gene ontology cellular component enrichment analysis was performed. This analysis showed enrichment in cellular components including ribosome, phosphatidylinositol 3 kinase complex, cytosolic small ribosomal subunit, extrinsic component of membrane, anchoring junction, focal adhesion, and transcription regulator complex. In conclusion, the study leverages CIDER to identify key genes involved in AML and utilizes various tools such as string, KEGG pathways, and gene ontology analyses to understand their potential roles and interactions within the molecular network. The findings suggest a strong connection between the identified genes and the mechanisms of myeloid leukemia, highlighting potential therapeutic targets for future research. Biological analysis of the TCGAL-AML dataset using CIDER revealed strong connections between key genes and related terms, including acute myeloid leukemia and the pathway in cancer. Over 40% of the key genes were linked to acute myeloid leukemia, with 15 genes also associated with colorectal, gastric, and breast cancer. Notably, patients undergoing chemotherapy and radiotherapy are at increased risk of developing acute leukemia. Enrichment analyses identified two significant subnetworks with dominant keywords, including acute myeloid leukemia and ribosome. The relationship between AML and ribosome is supported by research literature, which found that p53 pathway dysfunction is prevalent in acute myeloid leukemia. Specifically, RPL26, a ribosome gene, plays a critical role in inducing p53 and is connected to mTOR and other AML-related genes. RACK1, a gene associated with ribosomes, promotes THP1 AML cell proliferation by enhancing glycogen synthase kinase 3 beta activity. Overexpression of RACK1 is linked to chemotherapy resistance in T-cell acute lymphoblastic leukemia, suggesting that targeting RACK1 may be a therapeutic strategy for leukemia treatment. Similarly, the ribosomal protein S3 gene plays a significant role in acute lymphoblastic leukemia progression, and targeting RPS3 could be an effective therapeutic strategy. The pathway and term of focal adhesion also play a significant role in AML progression. CIDER identified important genes related to this term, and targeting focal adhesion pathways with specific inhibitors has shown promise in preclinical studies. Network analysis revealed a larger subnetwork including genes not on the KEGG-AML pathway, such as KIAA1549L, which has been implicated in acute leukemia development through its involvement in fusion genes. The CIDER method, a computational approach for identifying key genes and their relationships, was used for this analysis. Figures 3a-3d provide visual representations of the network analysis and gene relationships, highlighting connections between AML and ribosome, as well as potential therapeutic targets. Research has implicated the KIAA1549L gene in pediatric B-cell precursor acute lymphoblastic leukemia and adult acute myeloid leukemia, suggesting its role in leukemia development. Similarly, the CPNE8 gene has been found to negatively regulate cancer cell proliferation in acute myeloid leukemia. Analyzing key genes from TCGAL-AML data using CIDER has provided valuable insights into the molecular underpinnings of acute myeloid leukemia and related leukemias. CIDER, a novel method for causal subgraph inference, employs a counterfactual-invariant diffusion process to generate causal explanations, including reliable causal links. This approach enhances the reliability of causal links, making CIDER a general method for interventional causal inference applicable across various fields. A significant challenge in causal inference is handling unobserved confounders. CIDER addresses this issue using the task model phi, which splits observed effects into ESU and SO, assuming EC contributes more than EU. By removing ESU from EC, CIDER reduces the influence of unobserved confounders. The paper introduces CIDER as a method for causal subgraph inference, leveraging counterfactual and diffusion implementations to provide generalizability in evaluating a subgraph's causal impact on labels. Real-world evaluations demonstrate CIDER's advantages over existing methods, both theoretically and computationally. The counterfactual-invariant diffusion process is a key component of CIDER, enabling the generation of causal explanations and reliable causal links. This process involves a series of steps, including the identification of causal subgraphs and the estimation of causal effects. By combining counterfactual and diffusion-based approaches, CIDER provides a comprehensive framework for causal inference. CIDER's task model phi plays a crucial role in addressing the challenge of unobserved confounders. By splitting observed effects into two parts and assuming a specific relationship between them, CIDER can reduce the influence of unobserved confounders and provide more accurate causal explanations. The use of CIDER in real-world evaluations has demonstrated its significant advantages over existing methods, highlighting its potential for applications in various fields. Overall, The introduction of CIDER as a method for causal subgraph inference provides a valuable contribution to the field of causal inference. Its ability to handle unobserved confounders and provide reliable causal explanations makes it a general method for interventional causal inference applicable across various fields. CIDER, a novel framework for causal subgraph inference, is introduced as a model-agnostic and task-agnostic approach to identify causal relationships between variables in complex systems. By mitigating the effects of unobserved confounders, CIDER provides insights into underlying network-level patterns, addressing the need for explainability and transparency in black box AI. The framework's ability to infer causal subgraphs in real-world scenarios, despite unobserved confounders, is a significant contribution. However, further experimentation is needed to fully evaluate this feature and explore potential applications. CIDER consists of three main components: an additive noise model, a counterfactual-invariant, and a diffusion process. Trained using a graph neural network, GNN, based on measured data, x, y, the framework is designed to investigate the conditional distribution of edges, p, e, x, v, in complex systems and networks modeled as random graphs G equals x, v, e. The authors formulate the problem by defining key concepts and notations, highlighting the importance of graph neural networks, GNNs, and decision regions in predicting task-specific labels. The overall prediction GNN model is expressed as phi g equals phi fc, phi gc, x, e, where phi gc is the representation learning block and phi fc is the prediction head. The decision region of the task GNN is modeled as a convex polytope in Rho, encompassing graph instances with the same class label. Figure 1b illustrates the overall architecture of CIDER, which provides a detailed representation of the framework's components and their relationships. While CIDER shows promise in inferring causal subgraphs, limitations include the need for further experimentation to fully evaluate its capabilities. Future research directions include investigating causal subgraph inference in hypergraphs and exploring real-world applications. By acknowledging these limitations and opportunities for growth, the authors demonstrate the potential for CIDER to contribute meaningfully to the field of causal subgraph inference. The authors employ decision region theory to enforce constraints, segregating causal and spurious subgraphs, and compelling counterfactual samples to be as close as possible to the original data, as depicted in Figure 4b. This approach aids in distinguishing causal and spurious relations. The problem formulation is presented, where given a graph G comprising node features x, edges e, labels y, and a task model GNN y equals phi g. The objective is to identify the subset EC that has the most causal effect on y. If E is not available, an association-based graph of nodes can be constructed as an initial network instead. The authors focus on addressing the first question of explainability, which is to causally explain the phenomena itself, by formulating the problem as a causal effect problem. This entails learning a function f** operator that gives the causal subgraph EC, as defined in equation 3. The aim is to elucidate why y is causally affected by EC. To achieve this, the authors formulate equation 4, which emphasizes the causal relationship between the causal subgraph EC of E and the label y. They then define the spurious subgraph of E as S to represent the spurious subgraph of E, i.e., EC. To make the inference from association to causation, the authors formulate the problem using equation 6, which involves sampling a new S from the marginal distribution P, S, EC, without affecting the GNN's output. This condition is stronger than doing an intervention on EC. The counterfactual sample EC is defined as EC union S, as shown in equation 5. The authors emphasize that equation 6 is much stronger than equation 4, due to the docalculus or counterfactual implementation. If equation 4 is satisfied, GC is considered to only associate with y, rather than being the cause of y, unless equation 6 is satisfied. The application of structural causal models and additive noise models is crucial in understanding the relationship between signals and their noisy observations. An additive noise model, ANM, is a statistical model that describes this relationship, widely used in machine learning fields such as image and audio processing. The presence of noise in ANMs enables the identification of cause and effect signals. Counterfactual-invariant representation is introduced, where task-agnostic can be viewed as message passing. This perspective allows the problem to be formed as an out-of-distribution (OOD) detection task, aiming to identify a set of edges that can be removed without deviating from the original distribution. This concept is illustrated in Figure 4a, which depicts the method's directed acyclic graph, DAG. The distribution of causal subgraph is discussed, focusing on quantifying the causal effect or strength of EC. To solve this problem at the distribution level, the framework of random graphs is used. The relationships between P, E, P, EC, and P, S, can be formulated by EQ. 9. EC is a set of edges that induce transformations resulting in OOD of phi GC, X, when their distribution changes. Variational inference models are used to fit the distributions of E, EC, and S. This approach enables the estimation of the causal effect or strength of EC, which is essential in understanding the relationship between signals and their noisy observations. The use of structural causal models and additive noise models provides a robust framework for analyzing complex relationships in machine learning applications. Figure 4 illustrates the causal structure of CIDER, comprising a directed acyclic graph, DAG, where H is a direct causal effect on Y, and S represents spurious factors. The DAG is further elucidated through a simple O1 decision region of eCandison, a 2D space, showcasing different graph labels and sample types. The diffusion process for separating causal factors is visualized, with the red subgraph having a direct causal effect on the given label. To model the distributions of ACONDES, a variational graph autoencoder, VGAE, like model is employed, even in the absence of actual distributions or samples. This approach leverages the counterfactual-invariant representation to obtain a partial distribution of S without calculating the maximum likelihood of equal S. The objective is to satisfy equation 10, ensuring that the set S-hound in Figure 4a has no causal effect on under the condition EC. This is achieved by minimizing the total variation distance between the due distribution and origin distribution on road, as expressed in equation 10. However, exact calculation of the total variation distance is infeasible, so the L1 distance of the due distribution and origin distribution is approximately minimized, as stated in equation 11. Here, NC and NIS note the sampled numbers of the causal and spurious factors, respectively. By solving this problem, the subgraph of E that contributes the most to the label YCAN be inferred, thereby identifying the causal factors. This approach enables the separation of causal and spurious factors, allowing for a more accurate understanding of the underlying causal structure. The CIDER model is a two-channel variational graph autoencoder, VGAE, designed to simultaneously infer the distribution of the causal subgraph, EC, and the spurious subgraph, S, within a given graph. The process begins with an initial guess for the causal subgraph, which is then iteratively refined through a series of separation processes. This refinement process can be represented as a diffusion process, where the probability distribution of the causal subgraph at each step depends on the previous step's distributions. The CIDER model is trained using a loss function that combines four components. L1, which measures the causal effect of E on Y. LKLD, the KL divergence of the latent variables ZC and ZS with the normal distribution. LRE construction, the reconstruction loss of EC union S and E in the form of cross-entropy. And LTask, which prevents S from always being empty and E from always being equal to E. This multi-component loss function enables the model to effectively identify and separate causal and spurious subgraphs. The mathematical theory underlying diffusion processes is based on probability theory and stochastic processes, making them suitable for modeling various phenomena such as pollution dispersion, disease transmission, and information propagation in social networks. By leveraging these principles, the CIDER model can effectively identify and separate causal and spurious subgraphs within complex networks, providing insights into the underlying relationships between variables. This approach has significant implications for understanding complex systems and identifying causal relationships in various domains. The CIDER model integrates a graph autoencoder with a diffusion process in the hidden space to infer causal relationships between genes and cell types from single-cell RNA sequencing data. The graph autoencoder consists of a reconstruction loss term and a KL divergence term, combined to form the overall loss function. Additionally, a task-specific loss term is defined as the mean squared error between predicted and actual labels. The diffusion process in the hidden space is modeled using a Markov chain, where the hidden variable z is observed as a diffusion process. The transition probabilities are defined using the model's parameters, theta. By treating z as a diffusion process, similar to DALLE2, and leveraging edge reconstruction, the diffusion model becomes trainable despite the unknown noise distribution. The CIDER algorithm outlines the recurrent causal distillation process, where, at each step, ZCT and ZST are sampled from their respective distributions. The sampled values are then used to update ECT1 and ST1, which are subsequently used to update ZCT1 and ZST1. This process is repeated until t equals 0. The workflow of CIDER for inferring causal genes and cell types associated with a disease state begins with data cleaning on the original gene-cell expression matrix. The cleaned data is then used to infer causal relationships between genes and cell types. By integrating a graph autoencoder with a diffusion process in the hidden space, CIDER can capture complex interactions between genes and cell types, and infer causal relationships relevant to disease states. The model's ability to capture these complex interactions and infer causal relationships makes it a valuable tool for understanding the underlying mechanisms of disease states. The analysis process on biological data demonstrates the effectiveness of CIDER in inferring causal relationships between genes and cell types associated with disease states, highlighting its potential as a valuable tool for understanding complex biological systems. Data cleaning involves removing cells or genes with zero expression values, resulting in a selected gene-cell matrix, X-selected, a rectangular matrix with dimensions corresponding to the number of selected genes and cells. This matrix is then used to construct a gene network, E-gene, and a cell type network, E-cell type. The CIDER application is designed for two primary purposes: inferring causal genes for a disease status from single-cell RNA sequencing data and inferring causal cell types for a disease status from the same data. The CIDER workflow, illustrated in Figure 5, involves computing a gene-cell type matrix, X-gene, where each row represents a selected gene, and each column represents a cell type, with matrix entries being the average gene expression of a selected gene across all cells in a given cell type. By leveraging the gene network, E-gene, the gene cell type matrix, X-gene, and the task labels, Y, CIDER generates an explanation, EC-gene, indicating the causal effects of genes for COVID-19 and influenza. To infer the causal cell types, a cell type gene matrix, X-cell type, is constructed, where each row represents a causal gene, and each column represents a cell type. The matrix entries are the average gene expression of a given causal gene across all cells in a given cell type. By fitting the cell type network, E-cell type, the cell type gene matrix, X-cell type, and the label, Y, into CIDER, a causal cell type network, EC cell type, is determined, facilitating the analysis of the impact of cell types on COVID-19 and the combined effects of various cell types. This approach builds upon previous work on generating explanations for graph neural networks, as described in reference 1. I'm happy to help. However, I don't see any content provided for me to refine. Please provide the transcript you'd like me to refine, and I'll be happy to assist you in creating a concise, technically accurate summary suitable for voiceover narration. COVID-19 pathogenesis is a complex process involving multiple molecular mechanisms. A comprehensive review of existing literature highlights the crucial role of NADPH oxidase 2, NOX2, in COVID-19, with studies demonstrating its involvement in inflammation, oxidative stress, and cytokine storm. Targeting NOX2 may offer therapeutic benefits in COVID-19 treatment. The review also explores the relationship between COVID-19 and cardiovascular disease, citing evidence that SARS-CoV-2 infection can lead to myocardial injury and cardiovascular complications. Furthermore, the literature review discusses the potential role of ribosomal proteins in blocking SARS-CoV-2 replication and the application of brain disease network analysis to elucidate neurological manifestations of COVID-19. The Gene Ontology Resource and UniProt are highlighted as essential tools for understanding the molecular mechanisms of COVID-19. The prognostic value of S100A calcium-binding protein family members in predicting severe forms of COVID-19 is also discussed, along with the S100 protein family as therapeutic targets in pulmonary diseases. The complex interplay between various proteins, enzymes, and signaling pathways in COVID-19 is underscored, emphasizing the need for further research into the molecular mechanisms of the disease to inform the development of effective therapeutic strategies. The author's review of existing literature provides a comprehensive overview of the molecular mechanisms underlying COVID-19, including the involvement of NOx2, ribosomal proteins, and S100 protein family members. The review also highlights the importance of cardiovascular disease and neurological manifestations in COVID-19. Overall, the study emphasizes the need for continued research into the molecular mechanisms of COVID-19 to develop effective therapeutic strategies. Please provide the content that needs to be refined. I'll transform it into a concise, technically accurate summary suitable for voiceover narration, following the guidelines you provided. The relationship between acute myeloid leukemia, AML, and its potential link to previous diagnoses of breast, colon, and gastric cancers is a topic of increasing interest in the medical community. Understanding the incidence rates and underlying mechanisms of AML following these types of cancers is crucial for developing effective therapeutic strategies and improving patient outcomes. Research has shown that AML can occur as a secondary malignancy in patients who have undergone treatment for breast, colon, and gastric cancers. For example, Rosner et al. 1978 reported 24 cases of AML in patients with a history of breast cancer, while Valentini et al. 2011 observed an increased incidence of AML in individuals diagnosed with breast cancer. Similarly, Stein et al. 2012 found a higher risk of AML in patients who had undergone chemotherapy and radiation therapy for colorectal cancer. Wiggins et al. 1978 suggested a probable clonal origin of AML in a patient who had received radiation and chemotherapy for colon cancer. Zhang et al. 2015 reported a case of secondary acute promyelocytic leukemia following chemotherapy for gastric cancer. Kono et al. 1991 also documented a case of advanced gastric cancer associated with AML. In addition to the association between AML and previous cancer diagnoses, the paper also discusses the role of ribosome biogenesis factors in predicting the outcome of AML patients. Marcel et al. 2017 identified nucleolin as a novel potential marker to predict the outcome in AML patients. Furthermore, Quintas Cardama et al. 2017 found that p53 pathway dysfunction is highly prevalent in AML, regardless of TP53 mutational status. Zhang et al. 2017 discussed the role of p53 in myelodysplastic syndromes and AML, emphasizing its molecular aspects and clinical implications. Procosimer et al. 2017 explored the dysfunctional diversity of p53 proteins in adult AML, highlighting projections on diagnostic workup and therapy. Overall, the paper emphasizes the need for further research to understand the mechanisms underlying the development of AML in patients with a history of breast, colon, and gastric cancers. It underscores the importance of identifying biomarkers such as nucleolin and p53 for predicting patient outcomes and improving therapeutic strategies. Researchers have compiled a comprehensive dataset of 375 articles, comprising 10,012 samples, to investigate the efficacy of bio-inspired nanomaterials in environmental remediation. The dataset spans various pollutants, including heavy metals, pesticides, and industrial dyes, as well as diverse nanomaterials, such as metal organic frameworks, nanoparticles, and nanotubes. Principal component analysis, PCA, was employed to reduce the dimensionality of the dataset, identifying three key components that explain 83.4% of the variance. The results indicate that the adsorption capacity of nanomaterials is strongly influenced by surface area, pore size, and functional group modification. Further analysis using a support vector machine (SVM) algorithm revealed a robust predictive model, with a mean absolute error (MA) of 0.14, for estimating the adsorption efficiency of nanomaterials based on their structural properties. Notably, the model identified the synergistic effect of multiple functional groups on adsorption performance. Figure 4 presents a heat map illustrating the relationships between pollutant type, nanomaterial properties, and adsorption efficiency. The findings demonstrate that nanomaterials with tailored surface properties can selectively target specific pollutants, underscoring the importance of material design in environmental remediation. Limitations of this study include the reliance on reported data and the absence of kinetic parameters in the dataset. Future research directions should focus on experimental validation of the predictive model and the exploration of emerging nanomaterials for enhanced remediation efficacy. Overall, this research provides valuable insights into the optimization of bio-inspired nanomaterials for environmental remediation and offers a predictive framework for guiding future materials design and application. Graph theory, network analysis, and machine learning are interconnected fields that have seen significant advancements in recent years. This literature review focuses on graph neural networks, explainability, and robustness, highlighting their applications in cell biology, complex networks, and geometric inhomogeneous random graphs. The authors cite seminal works on graph-based methods for analyzing networks, including graph convolutional networks, graph attention networks, and the power of graph neural networks. They also emphasize recent advancements in robust and explainable graph neural networks, such as asymmetry learning for counterfactual-invariant classification, robust counterfactual explanations, and systematic evaluation of explainability methods. Techniques for certified adversarial robustness and robust learning with the Hilbert-Schmidt independence criterion are also discussed. These studies demonstrate the intersection of graph theory, machine learning, and biology, emphasizing the importance of understanding complex networks and developing robust and interpretable models for real-world applications. The comprehensive review sets the stage for the author's subsequent contributions to the field of graph neural networks and explainability. By summarizing key concepts, methodologies, and significant findings, this review provides a concise overview of the current state of research in this interdisciplinary field. Machine learning and artificial intelligence have increasingly been applied to environmental science, yielding innovative methodologies for understanding complex relationships between variables. For instance, Muij et al. 2009 proposed a regression method that minimizes the Kullback-Leibler divergence between the joint distribution of predictor variables and the target variable, facilitating causal inference in additive noise models. This approach enables the identification of causal relationships between variables, a crucial aspect of understanding environmental systems. In the realm of graph learning, Miao et al. 2022 introduced a stochastic attention mechanism that enhances the performance of graph neural networks by incorporating learned attention weights. These weights can be used to interpret the importance of different nodes in the graph, rendering the model more interpretable and generalizable. This advancement has significant implications for understanding complex networks in environmental science, such as pollutant transport and ecosystem interactions. In environmental science, Ukpaka, 2016, developed an empirical model to assess the impact of pH and conductivity on pollutant diffusion in soil environments. This study highlights the critical role of these factors in evaluating the potential risks associated with soil pollution. The model provides a valuable tool for understanding the complex relationships between environmental variables and pollutant behavior. The integration of machine learning and environmental science has led to the development of novel methodologies for analyzing complex systems. These approaches have the potential to improve our understanding of environmental phenomena and inform decision-making in fields such as conservation and sustainability. Further research is needed to fully explore the applications and limitations of these methodologies in environmental science. In the concluding section of the research paper, the authors delve into the implementation details and experimental settings for their proposed model, CIDER. The model's performance is evaluated on two datasets MUTAG and NCI-1, with their statistics presented in Table 2. These datasets vary in terms of the number of graphs, nodes, and edges, allowing for a comprehensive assessment of CIDER's capabilities. The experiments are conducted using a server equipped with an NVIDIA RTX 3080 GPU, 2.2 GHz Intel Xeon Gold 6148 CPU, and 256 GB RAM, ensuring efficient execution without excessive computational demands. The training process for CIDER employs the Atom Optimizer with a learning rate of 0.001 and weight decay of 0.0005. A batch size of 128 is utilized, and the model is trained for 500 epochs. A critical parameter in the CIDER algorithm is the number of diffusion steps, set to 10, which controls the depth of the diffusion process. Furthermore, the number of causal sample NCIs is set to 1, and the number of spurious sample NCIs is set to 4. The authors also compare their model against several baseline models, including Gene Explainer, PG Explainer, GEM, and Orphic X. These models are implemented using their respective official versions provided by PYG and the authors, ensuring a fair comparison between the different models.